So starch is a polymer formed from the monomer alpha glucose, and it is also classified as a complex carbohydrate. Starch comes from plants, and it is one of the primary ways that human beings get their nutrition on this planet. So let's take a look at the two different types of polymers in starch that can be classified as amylose or amylopectin. And the difference between the two has to do with the bonding between the alpha glucose molecules. So we're going to look at amylose first, and then we'll go on to amylopectin. So all glucose molecules in starch are alpha glucose. And we see here these two molecules at the top of the slide are both alpha glucose. The C1 position of each, the hydroxyl, is below the plane. And just remember, on the C1, it can be above or below the plane. In solution, that rotates. But all the other hydroxyls in the ring are set in their place for alpha glucose. So when amylose forms from glucose molecules, each linkage between two molecules is a condensation reaction, and then more and more and more and more occur to make a long, long chain. And by condensation, we mean two small molecules form a larger molecule, and a water molecule is released. Now, when starch forms, it always has some alpha 1,4 connections. And what does that mean? It means the hydroxyl on the C1 of the alpha glucose reacts with the hydroxyl on the C4 of another glucose. Now, in alpha glucose, that hydroxyl is always below the plane, and in glucose, the C4 hydroxyl is always below the plane. And so that makes an ether linkage between the two glucose molecules that is known as an alpha 1,4. That oxygen between the two monomers remains below the plane, and so it's alpha 1,4, 1, because it's linking the carbon 1 from one to the carbon 4 of another. Now, amylose only has this alpha 1,4 linkage, and it forms sort of a linear molecule that actually curls up and forms a helix. And this allows us to actually test for starch, because when iodine is added to amylose, it kind of nests inside that helix and forms a characteristic black color. Now let's look at amylopectin. Amylopectin absolutely has the 1,4 alpha connections. All starch does. But it has an added connection known as an alpha 1,6 connection. So let's look at these two glucose molecules on the top. As always, it's going to be the alpha glucose that forms these bonds. Here's our C1 with our hydroxyl below the plane, so that's alpha. Here's our other glucose, hydroxyl below the plane, so it's also alpha. What happens in amylopectin is a linkage occurs between C1 and C6. It is also a hydrolysis reaction where a water molecule is released and two smaller molecules come together to form a larger molecule. Now, what this does to the structure of amylopectin as compared to amylose is amylopectin has all of these branches due to this alpha 1,6 linkage. And what that means is it's easier for us to digest, because when enzymes start clipping these alpha 1,4 and these alpha 1,6 bonds to get glucose, they always have to start at the end of a chain. And since amylopectin has many more ends of the chains than amylose does, amylopectin is way easier to digest. Now, when we digest starch, what occurs, what the enzymes do is they facilitate a hydrolysis reaction where water is required and a larger molecule is broken apart into smaller molecules. And the very first step when we eat starch is we have some amylase in our salivary glands that is produced that starts to break down the starch. So if you ever eat some bread, this is often like a middle school experiment. If you eat some bread, you just wait a minute while it's in your mouth, it'll start to taste sweet because the glucose is being clipped off of the long strands in the starch.